हाँ, हेलो डियर स्टूडेंट्स, स्वागत है आपका रघुवंशी नीट के कैप्सूल कोर्स में। आज हम एक नया टॉपिक पढ़ेंगे और जिसका नाम है PCR। इट्स फुल फॉर्म इज पॉलीमरेज चेन रिएक्शन। It was first reported by scientist Carey Mullis. Now, what is the actual definition of polymerase chain reaction? That is clearly written: obtaining the multiple copies of gene or DNA of interest by using two sets of primer and the enzyme DNA polymerase in vitro. In vitro means it is performed outside the body in a lab. If you see the PCR, it has three steps: denaturation, second is the annealing, and third is the extension. Now, what are the components in the PCR? If you see carefully, it is a DNA sample, second is the primer, nucleotides, tag polymerase, that is a special type of DNA polymerase enzyme, mixed buffer, and the PCR tube. Now, this PCR tube carries all these components and they are placed in a machine that is called thermal cycler. Thermal cycler is an apparatus where you can fluctuate the temperature right from higher degree centigrade, that is from 94 degrees centigrade, up to the lowermost temperature around 40-30 degrees centigrade. So, before starting this topic, first we have to know what are the primers, what type of nucleotides which are used, what are tag polymerase. So, first take the primers. Primers are chemically synthesized oligonucleotides. Usually it is a DNA that is complementary to the parental strand. कहने का मतलब है प्राइमर एक शॉर्ट चेन ऑफ डीएनए है और उसके न्यूक्लियोटाइड्स पेरेंटल डीएनए के कॉम्प्लीमेंट्री होते हैं. Second, you are seeing the nucleotides. Nucleotides are present in the form of DNTPS. It means deoxy nucleosides triphosphate. As the name indicates, the nucleoside contain 3 phosphate group, and where the last 2 terminal phosphate is a high energy rich phosphate bond, and that provides the energy for polymerization of nucleotides. Third component is the DNA polymerase. And in PCR, a special type of DNA polymerase is used, that is called Taq polymerase. That was extracted from the bacteria Thermus aquaticus. Now this Taq polymerase, they even remain active at a very high temperature. So these components are mainly required. You are seeing also the buffer. Buffer help in the better functioning of the enzyme Taq polymerase. Now we have to see the different steps in PCR. There are 3 steps: denaturation, annealing, and extension. The first step is the denaturation, where in thermal cycler the temperature is fixed around 94 degrees centigrade to 95 degrees centigrade. Due to that, both the strand of the DNA get separate from each other due to breakdown of hydrogen bond between them. There I am showing with the diagram. Here both the strand of DNA having polarity 5 to 3, and this strand have a polarity 3 to 5. They get separate from each other. After that, the next step is performed, that is called annealing, where the temperature of thermal cycler is now lowered to the 55 degrees centigrade. Due to that, the primers which are present in the reaction, they start binding on the parental strand. So hybridization of primer with the parental DNA, this step is called annealing. Then temperature is again increased into this machine, that is up to the 72 degrees centigrade. Under this temperature, the third step progress, that is called extension. Now what happened in the extension? These deoxynucleoside triphosphate, which are arranged according to the complementary bases of the DNA, they are polymerized by Taq polymerase enzyme. And in this trend, also like this. So what here the Taq polymerase is doing? It is extending the primer by using the deoxynucleoside triphosphate in the reaction on the basis of parental strand. And due to this event, the one copy of DNA now become double. If this cycle is performed many times, then you can get the billion copies of DNA. For example, you can see here n is the number of cycle. If this cycle is repeated one time, the number of strands which are formed, number of DNA which are formed, they are 2. If this cycle is repeated like 2 times, the total 4 DNA will be formed. And like this, if this cycle is repeated many times, suppose 30 times or 20 times, then you can get the billions of copy. Now question is that why this PCR, या what is the application of PCR? तो for that जो है we have to study separately application of PCR. So of course the first application you have already seen, that you can get the multiple copies of the desired gene or DNA. The first application is the to obtain it have other also applications in DNA fingerprinting. Of course, during forensic applications. It is also used in the gene therapy experiments. And in molecular mapping. So these topics, that is the DNA fingerprinting, gene therapy experiments, and molecular mapping, you will study in another class. But here, the first, that is the most important thing, is that to obtain the multiple copies of desired genes. This can be used to study the mutation in the gene in a cancer suspected patients, or you can detect HIV viruses in a AIDS suspected patients. So for today only up to this you will study, and next class you will study about the restriction enzymes. Thank you.